with the little foldable picture. They're going to get taped together like this. And then I've added color so that I can easily see which components go with which. So starting up here in the space that I have, I gave myself an idea of what's actually going on here. So this diagram that we have here, it only represents half of the replication bubble. So this is the replication bubble, and the dotted line is showing this right here. So this foldable, this picture, is just showing this part right here. 5' to 3' strand on top, 3' to 5' strand on the bottom. This is where replication began, and it's moving in both directions. That's a pretty rough arrow there. Okay. There is a reason for this being a long, continuous arrow, and this being short, broken arrows. And here's another long, continuous arrow, short, broken arrows. So DNA A, that's a little subscript A, lowercase a, DNA A, the initiation protein recognizes the specific sequence that is the origin of replication. It binds and opens a bubble. So next are these different components that work toward making a copy of DNA. Helicase unwinds DNA by breaking hydrogen bonds between base pairs. And that's my green one, and I'll go through the colors and show you on there in just a minute. Topoisomerase is also called DNA gyrase, and so gyration has to do with turning. It relieves tension during the unwinding by cutting the covalent sugar phosphate backbone. Single-strand binding protein, SSB, they keep DNA strands from rejoining. RNA polymerase builds a short segment of RNA, and this acts as a primer. So there are 10 to 60 nucleotides in that short sequence. RNA primase, that primer, works from DNA's 3' to 5', so it builds a 5' to 3' segment. DNA polymerase 3, it can only add nucleotides in the 5' to 3' direction of DNA. It can only add nucleotides to existing ones, and so that's why we need the primer. And we kind of talked about that primer in the PCR reaction. You heat it up to almost boiling to unzip the DNA, but then we have to cool it down just a little bit so that that primer can bind. And the primer binds so that DNA polymerase is able to have something to build onto. Endonucleases, when the wrong DNA nucleotide is inserted, DNA polymerase 1 and 2 can remove the wrong nucleotide and replace it to fix the strand. So DNA polymerase 3 is building the big long strand. DNA polymerase 1 and 2 can go and fix any mistakes in that DNA. Nucleoside triphosphates, so just like DNA molecules, but they have 2 extra phosphates, and that gives them the energy that they need, and so for energy abilities like ATP. So phosphate hydrolysis means that you have energy to catalyze the synthesis of the addition of the DNA nucleotide. So nucleoside triphosphates have that energy to bond that nucleotide, to add that nucleotide to the DNA. DNA polymerase 1 removes RNA and replaces it with DNA nucleotides, so that RNA primer that gave DNA polymerase 3 a spot to add to, well, it's RNA. We don't want that in there. So DNA polymerase 1, it's one of the ones that help fix if there's the wrong nucleotide there, it also can remove that RNA primer and replace it with nucleotides. Ligase is glue. Ligase attaches Okazaki fragments together. It bonds the little nicks that are between them. All right, so helicase was green. Here's my helicase, and is working down to unwind, unzip, and break the hydrogen bonds between the two strands of DNA. The topoisomerase works to relieve that tension, untwist, by cutting those covalent bonds in the backbone. Single strand binding protein, so that kind of coats these guys, because remember, these have those hydrogen bonds and they want to stick back together real quick. We don't want that to happen because we have to make the new DNA. So it gets coated with this single strand binding protein so that they can't stick back together. So once we have these strands unzipped, then we need a primer in order to start building DNA. So here is my RNA primase. That's adding a primer here. So that way the DNA polymerase can bam, 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 add nucleotides. So number 5, my yellow one, is my DNA polymerase. DNA polymerase can only work in the 5' to 3' direction. So 5', 3', 5', 3' is going to be on the other side. On this leading strand, the DNA polymerase can just keep going and adding and adding and adding and adding, because this side's going to continue to unzip, and so we can just keep adding them in here. These are the nucleoside triphosphates, and so they are being added as it builds that DNA. On the lagging strand, it's being unzipped in this direction, and we need a primer. So my RNA primase adds the primer, but remember, DNA has to work in the 5' to 3' direction, so it has to build them from the left side to the right on the bottom. So I add a primer, and then I add them in. Add another primer, and then I add them in. Add another primer, and then I add the nucleotides in. So that's why we have these short segments, because as this DNA, it unzips a little bit, we can put a primer here and then build DNA. But then we have to wait for it to unzip more to add another primer and build DNA. And then we have to wait for it to unzip more to add another primer and to add DNA. That's what's creating these fragments, or these pieces. Those are called the Okazaki fragments. Ligase is going to bond together. It's going to glue together those Okazaki fragments so we have one long continuous strand. Let's see, we have DNA polymerase is right here. It would be working up here as well. This is DNA polymerase 3. This is DNA polymerase 1 because it's red. And it's popping out those RNA primers and adding in the correct DNA nucleotide. I gave myself one more little diagram just to be helpful. So on this situation, remember, this is the side that we're looking at here. So this is my leading strand, this is my lagging strand. Because on this side replication is occurring in the other direction, this side would be the leading strand on the right-hand side of the origin of replication. And because it's opening up this way, then we have to do the little fragments, and so this would be the lagging side. Leading or lagging has to do with, is it continuously being built, or is it being built in fragments?